a few things which I did for my research and as well as in the field of microbial ecology and also in general and also particularly in the water and wastewater treatment with the help of various types of microorganisms. First of all, let me thank to Professor Shagutla Madam for giving me an opportunity to share my research views with all the esteemed teachers and also MMTTC for inviting me as a resource person for this refresher course. My dear friends, just let us start with this my session with a small story, which could be a whether I don't know how many of you are familiar with this story or have all these aspects scientifically, whether you know or not. So let me start with that a small the story with that. So we are all familiar with this one of the epidemic diseases which is called as a cholera. Is it right? So we are all know how to control the cholera at present scenario, but whereas when it is an epidemic disease or when it outbreak has been taken in the particularly in the developing country like Bangladesh, there was a lot of debate was there how to control the cholera in the 1999. I'm talking about in 1999 when this outbreak has come up in the Bangladesh. Particularly when compared to the India, the Bangladesh is still it's a backward country because of facility for particularly in the hygienic conditions like drinking water facility and other aspects as the resources are uh, very in a small scale. During that conditions, particularly the water resources, which is used for the drinking purpose or also potable purpose, this was very a Herculean task for them. How to control or how to get this very clean water for the potable purpose or although as well as the drinking purpose. During that time, this story would will going to help all the publics which they had could able to find out how we can overcome for this problem. So that was uh, the framework of this story. So usually what we will do, so whenever the we will consider this water is contaminated or it cannot be potable or it cannot be drinking purpose, then what we will do, there are some traditional approaches we have, like we will going to purify with the help of filtration, and sometimes we will going to boil to remove this hardness or temporary hardness. And nowadays we are going for this RO filtration unit, and that is the individual level. But when it is a public level, in the corporation, what they will do, they will do the treatment process, like they will go for the coagulation, and after the coagulation, they will go for the sedimentation and the rapid sand filter. Then finally, they will go for the disinfection. In public, particularly disinfections in Indian country, I mean developing country like India, we have a few technology or a few approaches like using of ozone or treating the drinking water as a ozone treatment and as well as UV radiation treatment. These are all the very little bit costlier, but regularly what we are using is the sodium hypochlorite. That is what we are using, like that's chlorination method is familiar, which could be a very low budget. We can do that. So that is the main traditional approach what we are going for in public level. If something happens or something like even the contamination occurs, what we will do? Immediately, we will going to provide a medical care to the particular the person who has affected with the any types of epidemic diseases through the waterborne disease. Apart from that, we have lots and lots of the vaccination. What we have seen, and that will also give the vaccination. And also during the birth of the children also, we will give the various types of vaccination. So periodically, we have for various types of diseases. These are the some approaches. Apart from this approach, there was a new uh, thing which has taken up during the outbreak, which has done or which has happened at the 1999 in the Bangladesh, what I have already I given the that's the framework. Okay, so here during that 1999 there was a diarrheal diseases. So major cause of the death, particularly less than 5 years old, the children's, it has created a lots of massive loss of body fluid and also the electrolytes. And particularly the reason behind or the vector behind this is the toxigenic, the disease which is called as a Vibrio cholera. That's a virus which is a Vibrio cholera, which is a causative agent of the cholera, is which has occurred in the native environment or particularly in the aquatic ecosystem, or where the people or public used to go for the collect the water for drinking purpose. So where it is transmitted like anything. So during that time, there was a big question for them, because it's a, as I told you, it's a very, very backward country like Bangladesh. They don't have the access to the many technologies what we are using at present. Even though the India is a developing country, but we have a lot of technology or a lot of the techniques which is going to be used for the purification of water. Whereas in the Bangladesh country, that's very, very, that's a backward country. They are thinking what to do, because very poor people are living there. During that time, there was the discovery has been taken by, say, a common man or a, so is a national scientific foundation. The former president, she was there, the by name called as Rita Colwell. So I think if many microbiologists are there, they might be knowing about Rita Colwell. She has invented, she has come across this situation, and she tried her level best to how we can control this cholera. Okay, so she has used a simple technique which is called as uh, eightfold of sari. Particularly when the women, they will going to use the saris, different types of saris will be there. So she used that sari and she had folded 8 times, and you can see that as a tumbler, whatever the container which could be collected for the water. So that tumbler was tied up with this 8-fold sari. So they filtered the water. So what is the outcome is, so particularly the Vibrio cholera, which is worked as a commensal with the one of the zooplankton, megazooplankton or macrozooplankton, which is called as a copepod, which is attached to the eggs of the copepod, that will be controlled or that has filtered through this 8-fold inoculum. Okay. So in this, what you can see this the sari, the mesh under the microscope. This looks like such a mesh or with a smaller inoculum. This inoculum has filtered the copepod, which in turn has reduced the size of Vibrio cholera, which is attached to the copepod. That was the major the invention which has done, which has shown that, and they have published a paper like a simple sari cloth filtration of water is sustainable for continues to protect the villagers from the cholera in the mat lab. So here, about 30% of the people who were going to depend on this water, they have used this 8-fold sari to filter the water sample which I have collected from that particular the source of water. There, the results of the usage of this 8-fold sari has resulted in 60% reduction in the growth of the cholera. This is the so what we can see that even the small invention can bring a big change in a developing country like uh, a backward country like Bangladesh. This is one of the major invention which has took place. Why I am giving the example of this small story is we have studied this microorganism at both the level, like one is as a pathogen, another one as a useful bacteria or useful microorganisms. Because in the food, starting from the food, even in our body also, we have many different types of microbes we have. So that is controlling our body, even our pH level in the intestine, and also in our hands, and even everywhere we can able to find out this microorganism that are as a non-pathogenic or useful bacteria. So here I am concentrating on the usefulness of the bacteria. Apart from just I have given the example of small, the invention can give a big change in the scientific field. With this small story introduction, so I will give you a new concept which is going to be used in the field of a microorganism. As a zoologist or as a biologist, we all come across the ecology as a common in our studies. Okay. So whereas apart from that, there is a interdisciplinary subject which has taken birth in the 1984 or 3 decades of before. So there is with the help of SORB microorganism. That is nothing but a microbial ecology. Because nothing but ecology is whatever the biotic and abiotic factor which will going to be interact with each other, that will give you a definition of ecology. Whereas microbial ecology means in the smaller level or in the micro level, there are few type of microbes are there, which may be viruses, which may be archaea, or it may be a bacteria, or it may be protozoa, or it may be any type of microorganism which cannot be seen through a naked eye, but only observed through that's a microscope. So that how it going to interact with a physical environment, or how it will going to utilize the abiotic factor for their metabolic activity to carry out in the environment. That leads to a new concept or a study which is called as a microbial ecology. As a definition or scientific definition, we can classify, you can define as it is a study of microorganism and their relationship with one another and with the environment. Let us give an example of life. If so from the earth has come into existence, many death has been occurred. So what we will do, the dead bodies we are going to bury in the soil or ground. What will happen to these dead bodies means the microbes which will going to be degrade those dead bodies, and they will protect the earth from is not to be infected. Means the decomposition process is a major part of microorganism. That is how it will going to be have an interrelationship between the environment. Just I have given one example with the how it will going to degrade. Not only it degrades the human body, it degrades all sorts of the resources which will be buried or which will be there on the earth. Okay. So that is the main concept or main the function of these microorganisms, how it is going to be interacted with the environment. As I said, if they classify them, like we have an eukaryotes like archaea, bacteria, as virus, protozoa, and many such the organisms are there, which is going to play a very major role in this interaction. So particularly microorganism, which is also a play important role in the biogeochemical cycle. As a biogeochemical cycles, not only it will going to be focused for the particular place or particular the metabolism, it also occurs in various environments. We all might across, the familiar with the extremophiles. Extremophiles means the organism which is going to be living in the extreme conditions. What are those extreme conditions? like acidic lakes or the hydrothermal vents, and also in the deepest ocean where the lot of the salinity will be there, and also the human intestine, nose, and mouth. These are all the place where the extreme conditions are there. We can find these microbes in all such extreme conditions, and they'll perform uh, as a remineralization, as a remineralizer, as a vector of remineralization process that will occur all this in such a extreme environment. That is the main function of this microorganism. Let us the talk about this, how this concept has been occurred, because my title will going to be on the application of microbial loop in the purification of water and wastewater. Okay, so before going to the what is microbial loop, just I am giving you an introduction regarding how this concept has been arised. During the this metabolic activity, what I have told in the uh, particularly the biogeochemical cycle, here bacteria will going to function as a degradation and also transform an organic matter into the inorganic nutrients for as a food for the phytoplanktons. Phytoplanktons, here we can go for, if you consider or if you recall this, what is a food chain? Their phytoplanktons are become a producers there. So this bacteria will going to degrade those minerals and it gives as a nutrients to the producers, particularly a phytoplankton. Further, the bacteria serve as a food source for bacteriovorous or any other nanoplanktons. In addition to this, this concept has first they have observed in the marine ecosystem. Okay, so I'm telling that it's not occur in the freshwater, but whereas they have invented this concept or this microbial ecology is particularly in the marine. They have studied that how in the for a past 4 decades the microbial ecology has given the diversity of the microorganism in the various food webs, and as well as the complexity of the interaction and the metabolic the processes that derive from the biogeochemical cycle, that has given the concept of origin whether we need to study the importance of this microorganism in the aquatic ecosystem. That was the main the concept which has taken place. If we go to the origin of or a history of the microbial loop, so it not only started with the invention of the microscope, where the Dutch lens maker Antony van Leeuwenhoek, because without his invention, definitely we could have not seen how different types of microorganisms will occur, how it will perform, and what is the number. Even number will also play a very, very important role, because their abundance will going to help us the how much of the organic matter through the anthropogenic activity which will be released to the any nearby the water resources, and those microorganisms how it will go to degrade those organic matter. That matters a lot. That's why their abundance will also play a lot of role. So that abundance, if wanted to know that, the that's microscopic invention is the main important for us. Further, so in the 1900, there are lots of the developmental activity has taken place in the marine ecosystem. Even they studied the microbial loop at the marine biology. In addition to that, so there are the different types of the stratification which has occurred in the marine ecosystem, when we used to call as a hypolimnion, metalimnion, and epilimnion, like that. What we will going to stratification we will going to find out in the pond ecosystem. Similarly, in the ocean ecosystem also we can have a different stratification, where the bacteria not only lives at the bottom, it also have the different stratification based on their metabolic activity. So considering those things, they have a traditional the grazing food chains they have concentrated, and they have seen that what would be the impact of bacteria and other microbes, particularly the nanoplankton, which is going to help in the nutrient recycling. Nutrient recycling means, as I said, how it will going to degrade the organic matter into a useful nutrient to the producers. That will the process, that is the main functions of this microorganism. Whereas this microbial loop, the what we have wanted to tell you about, this is a concept in 1974 as Lawrence Pomeroy. He has observed that, so in the one of the food chain of ocean, where the microbes emerged as a major contributors to the carbon, that energy and nutrient cycling. So then further, he has observed those things and concentrated on that. So these microbes not only work as a primary producers, but also a leading consumers of the dissolved organic material which is released from the phytoplankton, or in particular, or in general, we can call them as a producers. So this microbes not only work as a primary producers, as a food for the nanoplankton, but also it absorbs the dissolved organic material from the phytoplanktons, okay, as a consumers. That was the main concept. So which has given a new terminology which is called as a microbial loop. Many of you have heard about this food chain. So that is in a higher level. We have a producers, consumers, decomposers. So as a food chain. So here the similarly a chain-like system which will occurs at the micro level, which is called as, which is defined as a microbial loop. Whereas in this microbial loop, the phytoplanktons act as a producers. Here, during their metabolic activity, the phytoplanktons, which will going to release the organic carbon, which is in the form of dissolved, and that's called as a DOM or DOC. It is a DOM is dissolved organic matter or dissolved organic carbon. This organic carbon, which is in the dissolved form, is a major food for the microorganisms for their metabolic activity. So that will be absorbed by the organic carbon. That's the microplankton, which may be having the 0.2 to 2 micrometer. That is called as a microplankton that will absorb the dissolved organic carbon. So they will do or perform their metabolic activity. During their metabolic activity, and as a next food chain, this microzooplankton will become a food for the nanoplankton, which will have a size of 2 to 20 micrometer. Whereas microplankton will have 0.2 to 2 micrometer. So this will be eaten up by the nanoplankton. Then these nanoplankton during their metabolic activity, or that will become a prey for the mesozooplankton or a tertiary consumer. Okay. So then this tertiary consumer, after the death of this tertiary consumer or a mesoplankton, they will going to release us the phosphate, nitrate, or other any minerals which will be utilized, or it will be transferred to the phytoplankton or picoplanktons, which will absorb as a food material. Okay, that is how the chain will complete. So that is what called as the microbial loop, as we have studied in the higher level of organism, which is a food chain. Similarly, a smaller the chain or a food transformation and remineralization, which all occurs at the micro level, which is regarded as microbial loop. This is the concept which has been first established by Pomeroy et al. In 1974. Further, this concept was further, the Azam, the person who is going to work on this microbial ecology, he took this concept and he's studied further, and he has clearly mentioned how the microbial loop works, like the dissolved organic matter released by the phytoplankton. So that will be consumed by the bacteria. The then bacteria will be consumed by the protozoa, then which are then consumed by the microzooplankton. That is a part of traditional grazing food chain. Okay, so thus this energy released as a dissolved organic material or dissolved organic matter by the phytoplankton, it's a rather the inefficiently return to the main food chain stream via the microbial loop of bacterioflagellate and microzooplankton. This is how the microbial loop has proceeded further by the Azametal in the 1983. So then you will have a question in your mind like, so much of things you are explaining about the microbial loop means what could be the use of microbial loop or what is the application? So why we need to study the microbial loop means because I will tell you that we are all in the present scenario, we are giving lots and lots of information. I mean, importance towards the climate change. And also we are blaming the various cities and human activity or anthropogenic activity leads to the climate change. But first of all, if at all you wanted to record how the climate change is occur or wanted to observe that, first we need to study about the microbes, how, because these are the organisms or the microbes which are the organisms which react first to the change in the climate. So that we need to study about that. So the one of the institutes which called as a National Center for Antarctic and Ocean Research, they are doing the research on this. Particularly, they are observing how microbes will react with when the climate change will take place. So that is why we need to study this microbial. Apart from that, it's not only just gives change or it will change according to the climate, but also it works as a remineralizer, and also it purifies the water. It purifies the running water particularly. So that concept I will come in the next stage or next slides. Okay. Apart from that, there is what would be the microbial loop means it not only the dynamic behavior of microbial loop, it not only works as just a food chain. Here in the microbial loop, we can see the whole several types of interactions, ecological relationship, like we have a commensalism, we have a computation, and also predation. So where the commensalism occurs means the production of dissolved organic matter from the phytoplankton and the utilization of the bacteria. This DOM, it plays as a commensal. Means the bacteria depends on the dissolved organic matter produced by the phytoplankton. That relationship is called as a commensalism. So here the phytoplankton will neither benefited nor harmed. Okay, that is how this relationship takes place. After the absorbing this dissolved organic matter, the production of the DOM or dissolved organic matter production influenced by the availability of the mineral nutrients. So how it will influence how much of our nutrients are available, that will matter to absorb that one. So the availability is very less. There starts the competition to absorb the micronutrients by the nanoplanktons, which is having the size of 2 to 20 micrometer. So there the competition occurs for the mineral nutrients between the phytoplankton and as well as the bacteria. So we can see both the commensalism and as well as the competition. Okay. Then this influenced by the growth of condition for the algae as well as the availability of the organic substrate for the bacterial growth, which is going to be observed or which is going to be taken up by the nanoflagellates or microflagellates. We can call it as a flagellate. We all know about what is the flagellates and what are the protozoans. We have flagellates and ciliates, both we have, and the protozoans. So there on the bacteria, which will influence outcome of this competition is the regeneration or of mineral nutrients resulting from predation will provide a feedback of some of the materials from within the microbial loop. Means once the dissolved organic matter is released by the phytoplankton, that will be absorbed by the micro organism, that is of micro level of 0.2 to 20, 0.2 to 2 micrometer. Then further, it will absorbed by the nanoplanktons having a 2 to 20 micrometer. Then microflagellates or microzooplankton, which will going to be used as a predator, and that will go, the nanoplankton become as a prey. This is how the different types of interactions which takes place in the microbial loop. So this microbial loop as a very important in the remineralization process and as well as a transform of from one stage to another stage. So how we can study these microbial loop, how we can know whether there any, because any sample we have given, or if you are subjected to any particular pond or a particular river, sir, can I see this, how the microbial loop will be there, or how microbial loop will occur, or whether can we study means. So definitely we can study that. So there are different types of techniques and methods are there. Let me talk about that also. So these are the traditional methods we have. Like directly we can go for traditional method like a plate counts, like agar plate counts, and we have a membrane filtration technique, the most probable number technique, direct more microscopic count, and dyes reduction test indicators, which will also use. Apart from that, that is a traditional method, and another like a rapid methods we have, because these traditional methods will takes play a time. Like you need to take the sample and dilute it and just pour it on. I mean, just put it on the agar plate, and you have to incubate for 48 hours. Then you observe under the the colony counter, you count it. There is a different types of colonies will come. Sometimes it's a colored and not non-colored. That colored is called as a chromogenic, non-colored is called as a non-chromogenic. So that will have the colonies. But when you study about the particular type of, if you take a streak or a small sample of that colony and you put it on the other slide and observe under the microscope, that will give you n number of different types of microorganisms. That takes time. But apart from that, there is another method we can call as a rapid method, where we can directly observe the microorganism under the microscope. That is called as a direct epifluorescent filter technique, where we will going to take the sample and we will going to add a nuclear stain, which may be a acridine orange or primuline, into the sample. Then you filter through the polycarbonate membrane of different background we have. White color background is also there, and dark color or a black color, the membrane filters are there. You can filter with the help of the suction pump. You take the filter paper, you keep it under the epifluorescent microscope, and you can observe the what type of, particularly this is for abundance, to observe the abundance, how many number of bacteria are there, how many number of zooplanktons are there, or how many number of protozoans, or any type of ciliates or flagellates, whatever that's there. So different techniques that we can use. That is rapid method, that's called. And also apart from that, electrical impedance, that's ELISA we have. These are the various techniques which is going to be used for to know about this microbial loop, whether it is occurring or whether it is there, how the bacteria will going to help us, and what number it is there. If it is number, it's reduced, how we can increase, because to degrade all the organic matter which is going to be released by the anthropogenic activity or industrial activity or any type of activity which is going to be polluting the environment, how we can culture those bacteria in the laboratory condition, how we can release it into the water to clean up that. That is how we can. As I said, this is a traditional method, plate count, and everybody knows. I don't want to elaborate on this. So I wanted to tell you about this direct epifluorescent filter technique, which is also called as a DEMT, direct epifluorescent microscopy technique, or also DEFT, or direct epifluorescent filter technique. Here, liquid food is what are the samples which can use is liquid food, then filter through the membrane. We can use acridine orange. As I said, it's the nuclear stain is there, acridine orange. That's very costly. When 10 grams, it will going to have you about 10,000 or more than 10,000. You can use that stain, a pinch of acridine orange you can take and dilute it in a known amount of water, and that stain which can be poured into the sample. whatever is a 10 ml or 5 ml, whatever you have collected from the particular sample, you keep it for 10 minutes, then you filter those water under the effluent, with the suction pump, we are using a 0.2 polycarbonate membrane. Okay. Then this is how we can count this manual or automatic. We nowadays, earlier, when just I'm talking about the 10 years back, we used to count this one with the help of the grids which is having in the microscope. Okay, that's called as a micrometry. So we used to count that. Even manual we can count, and automatic also it is there now available. Now it's connected with the computer. So you can just stretch the view which is there, which is automatically counted, and it gives how many number of bacteria are there. So there, the bacteria will see a different, the colors that will go. Here, particularly actinium orange is a fluorescent dye. You will observe the bacteria as a fluorescent colored, the microorganism. Okay. This is the technique which is I have told. So this is how we can go for that. So this is a filtration, the membrane filtration unit. So here, what you're supposed to do is, after keeping the funnel, you take the membrane of polycarbonate, you place it here, then fix the funnel, you pour the water sample which is going to be dyed with the actinium orange, then it will be connected with the suction pump, and it absorb all the water, because the pore size is very smaller. We can use the pore size here. They have used 0.45, even 0.22 micrometer is also there. For lower microbes we wanted to study, that you can do. Otherwise, that water will not be filtered unless you connected with the vacuum pump, because the pore size is very low. Then after filtering that, the membrane, you take out the membrane and observe under the microscope. This is the microscope which I have told. It's an epifluorescent microscope. So here the light source we have connected with the epifluorescent, the micros, that's a lamp. We have the different types of filters. We have a blue filter, we have a green filter. You can use the blue filter for the for count the microorganism which is attached to the camera. While observing itself, you can take them as many of photographs. This is the photograph which is going to see the different microorganisms, just like a stars in the sky. So you can, with the help of, like, these are the E. coli, Escherichia coli, and the various types of, even we cannot classify them by just observing. We cannot classify them, but the further the examination is necessary. Just I have given the photographs which is taken under the effluorescent microscope. This is one of the other things. So this is also which is taken in our lab only. So this is a sample of the Kaveri River. Just during our research, not only we have observed the microbes, or we have counted. Here you can see the fluorescent, the bacteria that's a rod shaped, cocci, bacilli, and also spring, the spiral shape, different types of the sizes. We can see that. Just I will going to just maximize that. You can see this, how it looks like, just like a stars in the sky. Apart from that counting, we also did the sizing. We also measured their size. Here we have used the graticules. In these graticules, we have measured their size. We have used this dark colored circles. This 10 dark colored circles we have. So it starts from a smaller range to a higher range. We used to move this micrometry, particular for under each bacteria. Whichever the bacteria fits under the particular circle, we used to measure that bacteria under those circles. That is how we did with the abundance of the microorganism through the epifluorescent microscope. So apart from that, as I said, there are different types of phytoplanktons also available in the water. So it will be astonished for yourself, like whenever you see particularly phytoplankton, you can get different types or different designs which can be observed of this phytoplankton structure. And not only the phytoplankton, we can also have some the euglena also there. So you can those things. So this is just a photograph which is going to done. These are the zooplanktons. So these are the zooplanktons where I have said this is the copypods. When they're all biology, particularly life science subject, you might have seen these kind of copypods, and also ostrocods, and also herpetocoids, and also we have rotifers, and we also different types of zooplanktons we can able to observe apart from this one. So these are all the things which is going to be observed for the particular water samples. As sometimes when you fix this water sample with the acridine orange, the bacteria will die and you can observe that. Sometimes during this abundance, how this bacteria, how the number will work means there is also one more the technology which is there, because uh, even when while counting, we will also count the dead bacteria also, because we doesn't know, because once we fix the bacteria or once we fix the sample, the bacteria will die. Even we'll count, we'll take into consideration that count also. This number of bacteria was there, and that can takes a degradation process that we will count it for that. But to see that out of the 100 bacteria, how much bacteria are active, so you can use this technique that is called as a bacterial activity measurement, where we will going to use this carbon 14. That's a carbon 14, that's a radioactive isotope that we will use. We will pour this carbon 14 into the water sample, and also we use some of the scintillation unit. We need to observe the radiations, how much. So that will be whatever the radiation that will occur, that will be trapped with the paper wick, which is going to kept in the bottles, which is going to be measured, and that paper wick will be closed or collected in a smaller vial within the scintillation floor, and that vial you can keep it under the scintillation liquid scintillation counter. So how much of the glucose uptake it takes place, which will be measured as a V maximum rate of uptake values will be observed, and we can measure that how much out of 1,000 bacteria, how much were active, how much were involved in the degradation process or purification process, that can be measured with the help of this activity. This is the one which we did. So just I will wanted to tell you that this is the river, the ecosystem which we have focused, particularly River Kaveri, and we have sampled all starting from the birthplace to up to the end of the River Kaveri in the Karnataka state, particularly. We are not concentrated on other states. We have collected all the samples with the different, and these are the few technique aspect which you have covered in the sample. Particularly what I told, it is abundance. Abundance is how many bacteria were there, whether it may be rod shaped, or it may be cocci, or it may be spirulae, or it may be any type of the types, or maybe immaterial. Only thing is we need to count our abundance. We need to observe in the particular samples. There we have classified into different types, like free living, which is not attached to any of the material, and also particle bound, which is attached to any organic matter. That's free living bacteria and particle bound bacteria. And we have also did with the activity, what I said is a Vmax by using a C14 glucose. And also we did a viable counts at traditional methods like colony forming units and chromogenic and non-chromogenic, colored and non-colored. And also we have studied a specific growth rate. Here we have taken the sample in the conical flask, observed, and taken the sample at the 0 hour, then 12 hour, then 24 hour, 36 hour. We could able to measure during these 46 hours, 0 to 46 hours, how much of bacteria has increased in the particular temperature or in the laboratory condition. That will give how we can grow those bacteria or helpful bacteria, which is used as a bioremediation process or a self-purification process, or anything that could be used as a degradation during the in the riverine ecosystem. For that purpose, we have studied. These are the few parameters. This is how, once again, which bacteria which we could able to see under the epifluorescent microscope. These are the phytoplanktons, which can, this is one of the indicator species we can see. It's called as a merismopedia. The species which is going to occur only in the water is contaminated with sewage. So you can see this phytoplankton. If the water is contaminated with sewage, then only we can see this kind of a bacteria and phytoplankton that's called as a merismopedia. So this is all the some of the flagellaria and the diatoms which you can able to see. So these are the zooplanktons. This is an nauplius stage of the copepod, and this is rotifer, and this is copepods once again. So these are different plights, and also we have published These are in the reputed journals, in the Journal of Bioscience and the Journal of Microbial Ecology, and also Indian Journal of Microbiology, which is going to be by the Springer. So apart from that, so here I wanted to focus on this particularly, where this microbial loop or microbial role will take place. Now, now it comes to the function of this bacteria. So I think you all heard about one of the proverb. We used to call our elderly person whenever you wanted to drink any water in any unknown place, or whenever you go to the visit to the place, if you wanted to drink the water for in a stagnant water, they used to tell, no, 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 don't drink this water, because the stagnant water will not having the purity. If it is flowing or it's a river in ecosystem, they will tell that, okay, you can drink the water because it's a flowing water. Means flowing water doesn't have any kind of a problem or any kind of a contamination. That's what that proverb in Kannada used to tell, that Hariya Neerige dosha illa. Means that Hariya Neerige dosha illa means that proverb will have the scientific background called as a self-purification or high biopurification. Means if the river ecosystem, if it is moving, the organic matter or the effluents or the sewage which is discharged by the human activity, that will be a particular a speed, a particular the distance. What it will do, it carries through the various, I mean, lots of oxygen will be there. That oxygen will going to help the microbes which degrade the organic matter which is released by the industry, or it may be sewage. So apart from that, whichever the higher particle or higher size particle that will be sedimented during the flow, apart from the sedimentation, the bacteria or algae which is there absorb the oxygen, they will decompose this one. And apart from that, we can also see that some of the biofilms, you heard about the biofilms, wherever the pebbles are there. So used to even filtration also, we have zooglial film or a biofilms we used to there. Means outer layer of this, the pebbles or this is covered over by the microbes, which is going to be initiate the process of the filtration or initiate the process of purification. That is how while moving the what dissolution of material and also polluted matters which is going to be settled at the bottom, and this biofilm will going to purify or degrade the organic matter which is released by the various types of industrial or human activity. And further, in the next or a downflow or the next the place where the water is moving, there all the organic matter will be so degraded with the help of microorganisms. This is the process which takes place during the self-purification. There particularly, they will have three different types of action. One is a physical action. What is the physical action means? It is going to purifying the effect caused by the hydraulic characteristics of the river water through the process of sedimentation, then filtration, aeration, bed load, and dilution. These are the process which is going to take place as a part of a self-purification. Okay. And also the apart from the physical action, there are both the physical and chemical action, which is going to affect caused by the flow and the flow rate. Means if the flow is more, you can get the more oxygen concentration in the form of dissolved. That is called as a dissolved oxygen. And this will have the contact with the river bed. As I said, the pebbles and the sand particles are a huge particles which is there. The material which will have the configuration like adsorption, aggregation. There, oxidation and reduction takes place as a biofilm act as a purifier there. Apart from that, the biological action means the purification action arising as a microorganism, means with the help of microbes, algae, bacteria, or any types of the living organism through various processes like adsorption, oxidation, decomposition, and synthesis of organic matter by the microbes of surface of contact media, particularly where the biofilm occurs, and also the taking organic matter and nutrient salts by the plants, and the sedimentation by the contact of each the microbes with the organic matter which is released by the anthropogenic activity. That is how the process will going to occur or takes place in the three different stages like physical and chemical and biological action. Okay. So here the actions which is going to involve in the purification in the particular streams or in the streams means one is a dilution. Dilution means whenever the organic matter which comes are discharged to the river, immediately what it takes, it diluted. It will be diluted means the more concentration of chemicals are there, it will be diluted through the addition. Then dispersion due to the current, and it will be dispersed because the water will be moving. Then higher or a larger particle will going to settle at the bottom called as a sedimentation, and also whatever the oxygen which is there, it takes or it going to be oxidize the organic or chemical content of which is discharged by sediment, effluents. Then reduction process, then temperature and sunlight are the major abiotic factors which will going to help the biopurification or self-purification in the stream, without which the temperature, if it is not there, that's why we used to call that as abiotic factor are very, very important. That is how the bacteria will have the interaction, that will have the connection with the how the abiotic factor will influence this decomposition process. That's why very much necessary is temperature, sunlight, pH are the main important abiotic factor which is required for to the purify that. This is about the riverine ecosystem or any drinking water. Okay. So if you come to the sewage, when the wastewater is there, we will classify as a water and wastewater. Water is for the potable purpose. Wastewater, we have two different types of wastewater. One is a sewage, another one is a industrial waste. Okay. In the sewage also, we have another type of water which is called as sewage. Many of the people doesn't know about what is the difference between sewage and sewage. Sewage is including with the human excreta, that is called as a sewage. The wastewater which without human excreta called as sewage. Okay, here the how the sewage will be treated means there the bacterial role will take place. Particularly, we will have a process called as an activated sludge process. Whenever the water is going to be released, wastewater, we have a bar screens, means any heavy particles are there or any the paper or wood or iron will be there, that will be filtered to the bar screens. Then water will be released to the next, where the grit or smaller particles will have the coagulation, will add the chemicals to sedimentation. Then the first sedimentation, after removing all the sedimentation of the heavier particle, we will send it to the aeration tank, where we will go to give the mechanical aeration and also the mechanical aeration. After that, even tube settlers are there. Many technologies which has come earlier, we used to have a fan-like structure on the top of the top aeration. Nowadays, it was tube settler, even bottom aeration or anaerobic digestion. We have many types of technology right now. So apart from after the aeration, we will give it a pond which is called as a secondary sedimentation tank, where we will add the digested sludge. Sludge means it is a solid part of liquid waste is called as a sludge. Okay. So liquid part of liquid waste is called as a leachate. Okay. Liquid part of liquid waste is called as a leachate. Solid part of liquid waste is called as a sludge. That sludge will have a n number of bacteria, n number of microorganisms. Those bacteria will be very active. We will take those sludge and we will add it to the sedimentation tank, where it will initiate the process of decomposition, which is even some bacteria will be very nullified or it will not going to work. So that will going to activate those microbes to activate or to for the degradation process. And over a period of time, detention period, maybe 48 hours or 48 days, that will going to clean all the organic matter or degrade the organic matter. Finally, we will get the clean water. So that water we can go for the further. If it is using for the drinking purpose, then you can go for the further like a biological treatment or the chlorination or a ozone treatment or a UV treatment or n number of types of the treatment systems are there. If that water can have or that we used as a drinking purpose. Okay. And you can tell that, you can ask that, sir, can we use this sewage water convert into this drinking water? Absolutely right. Even because of the using of this microbes, we can take out all 100% of organic matter which is already there in the sewage, that can be removed with the help of bacteria. That is the That is the application of this microbial loop. Means it want to take place means you should have the microbial loop. Because microbial loop means every component of the bacteria should be there in the chain. If every component should be there means you should have a abiotic factor which is supporting to this chain should have a proper temperature, proper sunlight, proper oxygen, proper minerals to complete this degeneration or degradation process. That is why it is our duty to look after the bacteria are good in that water. If it is there, then only we can able to get the very good water or in the lower stages or in the riverine or when it is starting from where the river will start and where it will ends in the downstreams. If it is degrading means the downstream people are highly benefited because of this the bacteria. This is how the microbial cell which will how it takes place mechanism is nothing but an enzymes which is going to be released by the bacteria or a microbes that will going to degrade this organic matter. Like the biological treatment we have, which will involve the use of microorganisms such as bacteria, viruses, and protozoans. They metabolize the biological content into the dissolved organic matter. That's, as I said, DOM. That's the sewage. Then the contaminants of the organic substances are digested as food along with other energy sourced by the cell. This is how just you can observe that only these three processes will make the water it's very clear and purified in the form of we can drink that water without any hesitation. That is how it takes place. For that purpose, I would like to give an example, like a living example of this Ganga. So many of us definitely, whenever we used to have the time or wanted to visit this place, Varanasi or a Kashi, we can use. Definitely, we will going to update ourselves. Like we need to take at least one holy dip in the river Ganga. Even after that, you do as for your information is concerned, about 25,000 tanneries are constructed along the stretch of the river Ganga. Tannery means the factory which is going to be used in the leather manufacturing. Okay. During the cleaning process, all these tanneries will release their effluents into the river Ganga. Apart from that, so every day, 1,000 to 2,000 unburnt, half-burnt dead bodies are thrown into the river Ganga. I'm not exaggerating. This is the present situation. But out of that, still we use this Ganga, particularly we used to call as a Ma Ganga, as one of the fragile ecosystem, and we used to take this water at the last stage of the human being. That's whenever the person who is dying, they will ask for that at least you put 2 drops of Ganga in their mouth. Why it's so? And why it is so fragile, or why still it's considered as a very fragile ecosystem means there is a virus called as a bacteriophage. It is not at all there in any of the other rivers which compared to the Ganga. Those are acting against the organic matter which is released by the different types of the dead bodies and also through the effluents to the various different types, particularly Kanpur. When we talk about the Kanpur stretch, you have more than 25,000 of industries are there. Now we have earlier, we used to 2014 earlier, we used to have a Ganga action plan. We have spent more than 2,500 crore. And now after the 2014, it has come NMCG, National Mission for Cleaning of Ganga. That's Namami Gange project has come up. Now more than 6,000 crores we are spending towards that. By involving all IITs, by involving all types of environmental scientists, which is going to be particularly IM Tech in Lucknow, IM Tech, Indian Microbial Technology Institute. They are also involved, and they have an astonished that still how it can occur. Even we can face or we can have this Ganga, how this self-purification or a biopurification is taking place. Why means is nothing but a called as viruses which are going to be eat the bacteria and as well as the harmful bacteria and also degrade the organic matter which is released by the various types of anthropogenic activity. That is one of the example. Apart from that, as I said, There is a recent the invention which has taken place, a new approach to clean the drinking water, just add a microbe. Just I'm not telling that in roughly or vague. It is one of the paper which is published by Meg Wilcox, a new approach to the wastewater treatment that could be cheaper to produce a less waste and possibly help to fix the nitrate pollution in the California and the sample. There you can add some microbes. What microbes? That you have to find out, that we have to work in the laboratory, and you culture those things in the laboratory condition. You, after the culturing, and you take that and conduct experiment in laboratory, and if it works, you take those bacteria and you release it to the water aquatic ecosystem. That itself will take place biopurification, or in generally what we used to call as a bioremediation, means the remedy to takes place with the help of living organism called as a bioremediation. In a bioremediation, we have microorganism we use, plants we use. That's as a phytoremediation we have. Like these microbes could be the one of the best tool for the bioremediation process, which is used for the biopurification or self-purification in the water or drinking water, whatever it may be. Sir. That is with these things. And we need to know that's what is good and what is bad. With these few things, I would like to